Thank you for the nice introduction. Hello everyone. Today, as you've already heard, I will tell you about HOGVAX. So this is a joint work together with my supervisors, Dilthey and Gunnar Klau. And here we are in the setting of peptide vaccine design. So how does peptide vaccines actually induce immunity? So we start with having a vaccine construct which we transport to, for example, antigen-presenting cells of the patient. And depending on how your construct looks like, for example, if it's given in mRNA sequence, it has first to be translated into a protein, and this protein is then cut by the proteasome into small peptides. And those peptides can then be picked up by molecules of the major histocompatibility complex. So those MHC molecules, they bind to the peptides and transport them to the outer cell surface, where they present them to T cells of the immune system. And if there is a T cell with a specific receptor that can bind to this peptide MHC complex, and additionally a co-stimulation signal is present, then this T cell triggers an immune response, and this in the end leads to the desired immunity. But this approach, so peptide vaccine, this approach comes along with several challenges. So first of all, those MHC molecules, they derive from the most polymorphic region in human DNA. So we have a high diversity of MHC molecules within a given population, but also across different populations. And also there is the phenomenon of linkage disequilibrium between different MHC loci, which you definitely have to consider when designing a peptide vaccine. And then those different MHC molecules, they have different binding affinities towards different peptides. So when you design your vaccine, it's likely that not each peptide in your vaccine is or can be bound by each of the possible MHC molecules. But even though that an MHC molecule binds and presents a peptide, that again does not necessarily mean that this is also recognized by T cells. And on top of all that is the challenge that peptide vaccines are limited in their size. And with only a limited number of peptides, you're not likely covering each and every individual of your target population, especially not the ones with rare MHC types, for example. So in combinatorial vaccine design, the general approach looks as follows. We start with a virus proteome, we cut it into peptides, and then in our case, we have an integer linear program that selects a set of peptides that maximize population coverage and also designs a single peptide sequence, which is then eventually translated into a codon sequence, which is then the part or the component of your vaccine. So in a basic approach, those selected peptides are simply concatenated or concatenated with spacer sequences in between, speaking of a string of beads approach. But as you can imagine, we have a limit, and when you concatenate those peptides, you exceed this limit quickly. So in our case, we're adding a little twist here so that we make use of overlaps between the peptides. So to say we are computing a shortest common superstring over the selected peptides, which compresses the vaccine formulation and allows us to add even further peptides to our vaccine formulation. And thereby we assume to also cover rare MHC types, for example. And of course, as we are computer scientists, we formulate this as a mathematical problem, which we call the maximum scoring k-Superstring problem. So we have a given set of strings, those are our peptides. We have a frequency vector f that tells us about the frequencies of the MHC alleles in our target population. We have a binary matrix B that tells us for each pair of MHC allele and peptide if they bind or if they do not bind. We have a parameter P, I come to this in a bit, and we have our length limit K. And now we are searching for a subset of our input strings, so a subset of peptides, P, for which we can compute a superstring S star with a maximum length of K, and additionally, the selection of peptides should maximize population coverage. And this is, so we say an allele is covered if at least P many peptides of our vaccine bind to this allele. So P is like a peptide hit parameter. So if we have those p many peptides binding to allele A, then our variable HA would be 1, and we are allowed to add the frequency of allele A to our overall score, and thereby we maximize population coverage. Now, if we find such a set of those strings, of those peptides, we compute such a string as prime, we call it a maximum scoring k-Superstring. So let's first have a look at a very basic example. So here we have a set of 4 strings. We have set our parameter p to 1. We have a length limit of k. We have a binary matrix where the columns correspond to the input strings and the rows correspond to the different alleles. And we also have a frequency vector. So now a very, very basic approach would be to just compute all possible subsets and compute the shortest common superstring for all of these subsets. And then just select the one that is within our limit of k and additionally has the highest score. So in this case, it would be the string SEETG. But you know, we have this shortest common superstring problem. We are also facing this set cover problem here. So this is an NP hard problem, and we need to think about how we can solve it efficiently. So what also comes into mind here is solving this problem using an overlap graph, especially when you think about computing shortest common superstrings. So we would construct an overlap graph by first of all inserting all our input strings as nodes, and additionally we add a start node here. And then we connect all the nodes with edges which we label with the number of characters we would have to add to our overall vaccine formulation when traversing this edge. So starting from the root node, for example, going to the string SEET, we would have to add 4 characters here. And now to find a maximum scoring k-Superstring, we would be searching for a cycle starting from the root node through this through this overlap graph, where the length of the cycle is at most k, and the visited nodes correspond to a set of peptides that maximize population coverage. But now we have a fully connected graph, right? And especially in our case, where we're dealing with multiple of hundreds, thousands of peptides, this graph exceeds our given memory quickly. But luckily, we found out about a data structure called hierarchical overlap graph, which was first proposed by Eric Rival and Bastien Cazot. And you can build it in linear time and space. And it is basically a compressed version of the Aho-Korosik tree. So it's a tree-like structure, and it contains all of our input strings, as well as all maximal overlaps between the input strings, and of course a root node that represents the empty string. Now we have 2 types of edges in this graph structure, and when you, for example, start at the root node and you walk down to the leaf nodes using those blue colored edges here, those are the tree edges, you pass all prefixes of the leaf node you're visiting. And those other edges, the red ones, they are called suffix links. They connect each node with its longest proper suffix within the graph. And now again, we are labeling the edges. So the tree edges are labeled with the number of characters we would have to add to our overall vaccine formulation. And the suffix links, they are all labeled with 0 edge weights. This is because the suffix is already included in the string we've built so far. And now to compute a maximum scoring k-Superstring, we would search for a walk through this hierarchical overlap graph, again starting at the root node. This walk should not exceed our given limit of k, and the selection of peptides we are visiting, they should again maximize population coverage. And here in this example, you can see those nodes with the cycle around it. Those are strings of the input set. And you can always walk back to the root node using a suffix link chain, so you can always go back to the root with 0 costs. And the hierarchical overlap graph is the key of our tool called HOGVAX to solve this problem efficiently. So here you can see the overall workflow of HOGVAX. We start with a given set of proteins, and then we cut the proteins into peptides of variable length. So for example, for MHC class 1, we consider peptides of length 8 to 10. And then we are additionally filtering those peptides so that we exclude, for example, self-peptides or peptides that are likely to mutate or peptides with a high glucosylation probability and so on. And from the remaining peptides, we build our hierarchical overlap graph. So then we decide for a target population for which we have the allele frequencies. Alternatively, you can also use haplotype frequencies here to take linkage disequilibrium into account. So we have these allele frequencies, and then for each allele of our target population and each peptide pair, we compute the binding affinities or the predictions for the binding affinities using tools like NetMHCPAN. And then additionally, in this case, we have to adapt those binding affinities to deal with our non-substring-free input set. So this is because we are using strings of variable length. And we are also assuming that our final vaccine sequence has to be cut into the different peptides. So we say that a superstring peptide is cut into its substring peptides. So we adapt the binding affinities in that way that a superstring gets the maximum score of itself and all its substrings as well. So then we also have our hit parameter P and also the maximum length of K, and all this is then input to an integer linear program that computes an optimal walk through the hierarchical overlap graph. And this walk then in the end corresponds to a final vaccine sequence and to a selection of peptides, and we call this vaccine formulation an overlapping epitope vaccine. So I evaluated HOGVAX using the SARS-CoV-2 case study proposed by Leo et al. And also compared HOGVAX to the tool OptiVax proposed in the same study. And OptiVax uses a beam search, so this is a heuristic breadth first search, to select an optimal set of peptides. Here the following computations are all done on single MHC allele frequencies, and I let OptiVax run first to select a set of 19 peptides for MHC class 1 and 2 separately, and then I concatenated those peptides to get our maximum length limit of K for HOGVAX. And within this limit, you can see in the middle plot, that by using the shortest common superstring approach, we are able to add significantly more peptides to our vaccine formulation. And of course, this also has an effect on the population coverage. So here I evaluated both tools using the evaluation function also proposed by Leo et al., where I compute the probability that each individual of the target population presents at least one peptide at any loci. And you can see that with this higher number of peptides, so in the right plot, we can increase the population coverage, even though that both tools are already like in the upper 90%. By following those superstring approach, we cover, for example, more rare MHC types as well. And this higher number of included peptides also has an effect on the robustness of the vaccine. So a vaccine is considered to be robust if multiple peptides of the vaccine are presented by each individual, so that in case of a virus that mutates, that if, for example, one peptide included in your vaccine is target of such a mutation, there are still other peptides that lead to immunity. And here I computed the probability for at least n many presented peptides per individual in our target populations, and here I'm using haplotype frequencies for the evaluation, while the computation still took place on single allele frequencies. And the haplotype frequencies, they derive from typed individuals of Asian, African, and European ancestry, as well as the average over these three populations. And what you can see is that both tools, OptiVax and HOGVAX, perform quite well. So for at least one peptide HA hit, we reach like 100% population coverage. But when you increase the number of required hits, then you can observe that OptiVax decreases much faster compared to HOGVAX, while with HOGVAX, we still achieve like 50% population coverage. And now you might remember this parameter P. So our hit parameter, if we set it to, for example, 10, we're able to create highly robust vaccine formulations. So here in this case, I set it to 10, and you can see that we achieve like 90% population coverage for at least 10 required peptide HLA hits, which is quite amazing, I think. So this already brings me to the conclusion and the overview. So I've shown you HOGVAX, the general workflow, that we start with proteins, we cut them into peptides, we create the hierarchical overlap graph, which is key of solving our MSKS problem within minutes. So even though that we have hundreds of thousands of input peptides, we are able to compute a peptide vaccine in like 15 minutes or something. I've shown you that following the superstring approach, we are able to add significantly more peptides to our vaccine formulation, and that in general, we are covering more than 90% of the population. And currently, we are also in collaboration with the lab of Philipp Lang at the University Hospital in Düsseldorf to test our approach in wet lab experiments. So this is because we are not quite sure if this proteasomal cleavage process really works as we assume it to work. So we're testing it for the LCMV in mouse cell line experiments. We computed a vaccine formulation with HOGVAX, and they designed a plasmid vaccine with it. And then we will see if the overlapping epitope vaccine is cut correctly, or if our approach will fail to unpredictable proteasomal cleavage processing. And with this, thank you for your attention. If you have any questions, feel free to ask. And also, I'd like to thank the RECOM again for the travel fellowship.